and also Myra, and also Kutonala. And right now, as Carla say, I really want to try to explain everything about vaccine and also coronavirus disease. And I want to say I am not very dominating English perfectly because I only spent few years in US, but I can try to share my presentation. Okay, I want to start with explain everything about what is COVID and also what we know about for the vaccine and also for the coronavirus. Okay, the title for this presentation is COVID-19 vaccines. And do everyone know the coronavirus is not really very new diseases. Also, coronavirus had the capacity to infect animals and humans. They divide in 4 different groups: alpha coronavirus, beta coronavirus, delta coronavirus, and gamma coronavirus. The virus is coming from the common ancestral. These are bats. The bats it's possible to infect directly to the human and also animals, or they use a host intermediary to cause several diseases. For example, pain, on avar, or camels, or cough. The human coronavirus is not really new. Actually, we know other human coronavirus to cause, for example, the common cold. The first 4 human coronavirus do we really know? I can show here in green color. The first one is coming actually approximately 50 years ago, and they represent 50 to 20 common cold. But the more important is the last 3 viruses to cause an epidemic. The first more important was the SARS-CoV-1, who is coming or was identified in 2002. It actually was in the same country, in China, in the Guangdong, and also in Hong Kong cities. And after that, 10 years after, is coming other new virus. The name is MERS coronavirus, to emerging in Saudi Arabia in 2012, and it's coming for the camels. And recently, everyone know about for SARS-CoV-2, who is responsible for cause the COVID-19 pandemic in 2019. And then I say before, this coronavirus is a zoonotic disease, and the common ancestor is a bats. For example, the 3 epidemics or pandemic is coming for the host intermediary infection. For example, for SARS-CoV-1 was a civet. For MERS was a camels. And for the SARS-CoV-2, we don't really know exactly what is the host intermediary, but according to the sequence, to several groups sequence the virus, the complete virus, especially in the S protein. We maybe know is coming for the pangolin, but it's not really 100% because the homology for the sequence is only approximately 96%. Something about for this, or what is specific or for these viruses. One characteristic is this virus is the very big virus. They had a very big genome. Approximately is around 22 kb positives is a positive polarity RNA genome. Here I can show what is the proteins for this virus. For example, the 2 ORFs, 1A and ORFs 1B, they divide in 14 non-structural protein. The name is NSP1 to NSP14. One characteristics for this NSP protein is they have the capacity to correct the the error during the replication, and what is the protein to have this function is the NSP14. I cannot explain all these proteins, but I want to say to the more important protein for example, for the vaccine is the S protein. The S protein is because it's the name is coming for the spike protein or the spike protein, and this is responsible for how the coron in the envelope protein. And what is the more important thing to know about the S protein is this protein is really the responsible for the immunity, and also I think everyone know right now to all the companies to produce or to make or to develop vaccine use the S protein to generate the immunity. Here I can show how is the structure for the S protein. You see here in the right low, here is what is the structure for the S protein, for example, for the SARS-CoV-1 and SARS-CoV-2. And this is responsible for bind when the AC2 protein to this is the receptor to is coming for the cells, and they is bound with the S protein. I mean, is the same to when we say how is the virus introduced into the cells is when S protein bind to the AC2, and this is the reason for go into the cells and after replicate the virus and produce more variants. Now I want to more explain what is the vaccine. Recently, I think approximately 2 weeks ago, we have published one publication to which showed several platforms to develop COVID vaccines. You see here is the SARS-CoV-2 variants. I show here the S protein. The several companies use this protein to make all several different platforms to produce vaccines. For example, the more principal is with DNA, with mRNA, vector to is dividing two, replicating or not replicating. And also we use only the protein, or we use the complete virus, but we inactivate the virus, for example, with chemicals or other things. Or we use the light attenuate virus. How we may, I explain later in this presentation, and other, for example, VLP. Very, very, I mean, very, I want to explain. I think I can. Why is the reason for I showed this presentation is because some anti-vaccine people, they use to this vaccine is no good because they produce in very short time, and another reason, right? And the reason for I showed this slide is because normally for produce or for make one vaccine, the time is around 8 to 15 years or more. What is the steps for produce this vaccine is I can show right now. For example, the first one is design and exploratory when I work in the lab with tissue cultures, with animal model, for example, more with the mice, and after with primates. See, everything is okay. After 1 or maybe 5 years, we pass with other steps. We test the vaccines in humans, and these clinical trials we divide in 3: phase 1, phase 2, and phase 3. Normally, in every phase, the time is during 1 or 2, 3 years, and after that, if everything works, we pass with review and approve. When, for example, in several agents, for example, FDA is the Food Drug Administration for US, and also for the EMA, to is the European Medicine Administration, yeah, I remember. This is the organization to to approve the drugs or vaccine, for example, in European Union. And also, for example, in Mexico is the Covide agency. And after is the manufacturing and marketing. And after that is the distribution. So this is a big problem, for example, in countries do not have very high economy, because some vaccine, for example, mRNA vaccine, they need very low temperatures. Finally, the administration to humans. So, but for example, why is the COVID vaccine they produce in very, very short time? Because also the first vaccine to administer in the people was approximately 10 months or less, because the first was in the beginning to the 2021. It's because I want to summary here, is because we know several information about SARS and MERS. So I say before, this is other human coronavirus we know, and also we know more about from other coronavirus to infect animals. And also before to finish this design and exploratory state, we start with preclinical, for example, in primates. And the more important thing is when we are working with trial clinicals in humans, finally, we did the overlapping phases. The meaning is before I finish the phase 1, we start with phase 2 or we start with phase 3, and use also before to publish, for example, right now, before to publish some vaccine with phase 3, they start to authorize or also administration. And another important is to the companies or the pharmaceutics produce the vaccine before to have the authorization. And this is very not very common, because imagine they produce thousand and thousand dots, but finally they not accepted for example, FDA. They lost a lot of money, especially dollars. And also after with the administration. This is a specific reason for why the COVID vaccine way produced very short time. And also the right now the mRNA methodology is very recently. So it's around, not very new, but it's around 20 years old. They produce other vaccines, for example, VIH or cancer vaccine with this methodology. This is all a novel coronavirus candidate developed in the world. You see, for example, in the clinical is more to 184 vaccines right now. So right now it's more to 200 vaccines in study. And you see here, for example, is 18 vaccine is authorized. And I want to say something important, because the vaccine is very safe and it's very efficacy, because not all vaccines to study is really authorized. For example, here in green, I showed for the moment is 17 vaccine is not approved. So the company lost money in this case, and they start to produce other vaccine, for example. And this is the reason for all the vaccine we are really have right now in the world is very safe. I can show why more later. And this is other only slide to show what is the COVID vaccine candidates in clinical phase. It's more to 100 right now. And here is, I summary in this table, what is the more advanced vaccine for dependent for the platforms. For example, for DNA is Sidus cadila is in for India. For example, mRNA, we know perfectly what is, for example, Pfizer or Moderna. And it's other vaccine, for example, CureVac, but it's not very good result right now. Inactivate, for example, Sinovac or Sinopharm, or light attenuated is Institute for India, Codaxin. The more for the people to remember is, for example, viral vector vaccine is, for example, AstraZeneca for Oxford, CanSino for China, for many people, for teachers for Indian university, they receive or elementary school. Cabalenia to is the Sputnik V for Russia, another one. For example, Johnson & Johnson for US. When we talk about for vaccine, we want to know how is the immune response work when the viruses. For example, when a virus had the contact with, for example, with humans or with the cells, they produce principally B cells. To these B cells, the function is neutralizing antibodies and Ts. With that, we prevent the infection and also produce the T cells, especially CD8 and CD4 T cells, and they make cytotoxic T cells, and the function is destroy infected cell and finally protect the several infection. And some cells very important is the long life memory. So this is responsible to have the protection, but for months, for example, or for years. One problem right now for the vaccine is we really don't know what is the time to protect for the people. For example, many studies show to 6 to 8 months is okay, but we really need not to the dose is worse during 1 or 2 years. This is the reason for recently in the war, some countries decide to have the additional dose, for example, the 3 dose, for example, for Moderna or for Pfizer, especially in Euro Union or US, for example. In Mexico, right now is not the time. How is the vaccine developed how vaccine develop immunity? This is very summary how is the COVID vaccine works. When after they administer to the people, they show the antigen present cells, for example, in dendritic cells, and they produce when the time normally they start 1 or 2 weeks after the dose, and normally when it's a complete scan after the second dose, we need to wait minimum 2 weeks to have a more good immune response. And then I say before, they produce especially 2 ways, B cells and the T cells, or to produce antibodies or CD8 and CD4. But what is the efficacy for the more relevant or the more important vaccines? For example, we want to define something here to normally the efficacy represent to symptomatic cases or symptomatic cases. For example, the majority is between 94 to 60 is the efficacy, but this is to prevent a symptomatic case. But it's different when we want to say what is the efficacy to prevent hospitalization or pass away. For example, the majority is around to 85 or to 100% in the majority of the cases. Also, something to have important is the common adverse effects after the COVID-19 vaccine. This is because this is important because sometimes for no very good information, people don't want to receive the vaccine because they think into this is had several common adverse effects. Here I divide, for example, in minors and non-minor. For example, I show here the minors. The minor is, is the more common is the fever, pain, injection site, fatigue, headache, or nausea, or muscle pain. And in the majority of the case, depends to what is the Moderna, Pfizer, but normally in all vaccine, you have the minimal common adverse. Not all people have these effects, but this is depends for what is the immune response also for the person and also for the age. Other thing important about for the vaccine is, for example, how is the T cell response in recovered patients with COVID-19 disease? We know when the people receive the vaccine, they had immune response, but also when the natural infection, the people had immunity. I mean, not only the neutralization antibodies, but they also produce CD4 and CD8 T cell response. And also in this publication recently showed to other proteins is implicated in had other viral and had other immunity. I mean, I want to explain the other way, for example, right now, all the vaccines produced or developed with the S protein. But right now we know for example, the membrane protein, the nucleocapsid protein, or the non-structural protein had a role with immunity. And in the future, we want to produce other vaccines to have other targets, for example, N plus N or S plus N, in order to have very better vaccines. And how is the RNA vaccine works? First, we have designed the DNA vaccine is a template, and we have put what is the correct sequence, for example, for the S protein. And after we have the in vitro transcription. After the vitro transcription, they make the mRNA. This mRNA is naked. We need to protect because it's degraded. And for this, we have had the formulation, and after the formulation, they administer it to humans, and they produce the antigen and antigen protein reaction. Here I can show how mRNA vaccine works. And I say before, after we have produced the RNA, for example, the S mRNA, we are protected with the lipid nanoparticles, and these lipid nanoparticles introduced to the cells to have the S protein expression. After this protein expression, they made the antigen presenting cells in dendritic cells. These dendritic cells express the helper T cells, and they also produce making antibodies to neutralize the virus and also killing the infected cells. This is how is normally the vaccine work. And something to consider is to this vaccine is not integrate to the genomic DNA of the cells, and also is complete conservation and distribution. Something to consider for this vaccine, the advantage is because this is very safety, had very good efficacy, and also the large scale production is very easy to produce a lot mRNA vaccines. The challenge for this vaccine is stability in the cells with modified, for example, nucleotides with pseudouranilos, or reduce the amount to GC when in cytosines, and to study several delivery systems, for example, use different polymers or stability, because right now the mRNA vaccine, we need to stay in -80 or -70s. But for example, in Africa, for example, today don't have freezers. This is important to have vaccine we are concerned in room temperature, for example. This is the challenge. Here I show, for example, what is the chemical to use for majority delivery methods for mRNA vaccine. For example, we can use protein, liposomes, or cationic lipids, or electroporation or protamine. And also, recently, last year, we have published other publication to show how the nanomaterial, how use the nanotechnology to produce vaccines to generate more activation for the human system, and also to use this nanotechnology to generate more nanomaterials, for example, mass or drops on other things. I show here this picture because I want to say to generate vaccine or to work with this virus is not very easy, and not all the laboratories or not all the countries have the capacity to work with that, because we need especially especially laboratories, and the name is Biological Safety Levels Laboratory, BCL3. Normally, the laboratories to work in universities or in these institutes is in the BCL1 or BCL2, but to work with SARS-CoV-2, or for example, with other pathogens, for example, Ebola, we work to have in BCL4 labs. and also the person to work in these labs, we need to have very careful. I mean, they need to work very careful, have experience. They have training, special training. I know I want to show only these photographies. For example, here we have worked with animals, for example, with mice, to test, for example, the efficacy with the vaccines, for example. I can show here how we are made, for example, light attenuated viral vaccine. For that, we use, for example, two approach. The first is infection clone is the full length cDNA. Another is the animal models. For the animal models, for example, we use normally mice. The normal mice, I say wild-type mice, normally is not susceptible to the SARS-CoV-2 infection. For this reason, we need to make special mice or modified mice or transgenic mice. To the name is mouse adapter SARS-CoV-2. For example, their characteristics for these mice, they need to have the high titers in the lungs after the infection. They need to produce viremia, extrapulmonary spread, neutrophilia, pathological change in the lung, and also pass away for tests, for example, antivirals or the vaccines. What is the mechanism or the pathogenesis to produce, for example, viral attenuated vaccine? For example, we need to identify what is the virulence factor in order to delay several genes responsible for the virulence. But these genes, they don't need to have the essential for viral replication in cell culture involving virus virulence in vivo interference with the innate immune response. And if this delayed CDIPS gene after deletion is cause attenuation, this is maybe possible to use as a vaccine. I explain in the next slides. For example, in this slide, I show how some years ago, we are in my previous lab, we are make one vaccine for SARS-CoV-1 by genetic engineer. We modify the genome. We are delayed two genes important for the virulence. For example, the first one we delay is one small part for the NSP1 to is no structural protein. We are delayed this part. And also we are delayed one small part for the envelope protein, for the E protein. And with this reason, we are testing animals and also in tissue culture. And we want to note that this modification is very good vaccine candidate. In order to test if this vaccine we are developed, we are test the clinical, or we analyze the clinical disease and lethality in starting way in mice or survival. We had three different groups here. In green, the mice we infect with amok, for example, with media, and also the black groups we previously infected with the wild-type virus with SARS-CoV-1, and also in green, in other, the three groups was infected with the vaccine candidate. And we can show here to only the mice we infect with the wild-type virus, with the black, they lost weight, and also all the mice die after 5 days. So with that, we can say to the mice weight infected with the negative control, or when we previously infected with a vaccine candidate, no lost weight, and also the 100% the mice survival. So with this reason, we can conclude to this vaccine candidate has an attenuated phenotype. But something we need to analyze when we test with the vaccine is how this vaccine cause the pathology in the lungs, because we know these viruses are respiratory viruses, and they cause problems or change in the lungs. For example, this is the lungs after 2 days post-infection or 4 days post-infection. you see how is when we infect with a negative control with the moth, or how is the lungs look when infected with the SARS-CoV-1. This is, you see here is water. Is said the name is edema, and when this edema, this is cause a very contraction in the tissue. But this is some reason for we cannot have the intercurrent for the gases in the tissue. But for example, if you see how is the vaccine candidate after the vaccine candidate, it's very clean here. Is no edema. When this result, we can say to this vaccine general fully attenuated virus that is not induce significant lot damage during infection. But what is the really function here for the vaccine? The vaccine is how is the function, how is work the vaccine? Normally, people they know exposed previously with the pathogens, they administrate the vaccine and they wait around 2 weeks or 3 weeks after we generate antibodies, T cells. And in the case to the people exposed to the pathogens, we had the defense to combat or neutralize the pathogens. For the reason we use or we make one experiment to challenge win the parental virus. I can explain here is the immunization. First, we have two different groups. One group to we administrate the vaccine, another groups to not administrate the vaccine. After 21 days post-vaccination, we challenge the mice with the parental virus, with SARS-CoV-1, in order to know to the vaccine generate antibodies, T cells, and protect the infection, right? So we analyze the clinical disease, lethality in the way, and the survival in the mice, but you see clearly to the mice to not receive the vaccine, they start to lose the weight, and also all the mice die, but not the mice to will receive the vaccine. You see, this is very clear to we are generate one stable attenuated viral vaccine induce full protection in mice. So for the timing, I cannot show more experiment, but we test the vaccine in several mice to have young mice, old mice, or more time to, for example, we test in 40 days post-vaccination, because it's important to know for how long is the protection for how long. So I go more because more faster. Something important to consider here in immunity is the natural infection. Normally we make two different immunoglobulins, the IgA and IgG. For example, when we receive the vaccine is for intramuscular vaccination. Normally, the name is not sterilizing immunity because they have the systemic and virus replication, and they may only, or the majority IgG systemic. For this reason, when we are receiving the vaccine, we are protected for hospitalization or for the pass away, but we continues to transmit the virus. But one idea to avoid to the people to have the vaccine and not transmit the virus to other persons is to produce IgA, because they had immunity in the, for example, in only this part in the upper respiratory, I think, tissues, and for this idea, how we can produce this immunity in IgG is with intranasal vaccination, and many companies work with make vaccines by intranasal administration. With that, we produce a sterilizing immunity in the mucus and cause the virus not replicate, and finally not pass the virus to other people. Sorry, I saw to Stephanie had one question. Maybe later.
Yeah, maybe because.
Yeah, sorry, it's because I saw the hand, but I want to finish the slide. Okay, another things to consider when the vaccine, when the SARS-CoV-2 is the efficacy, because the efficacy maybe when the time reduce the viral transmission, the variants we know really right now to every time you see in the news to is new variants, the Indian, for example, right now, the more important is the Delta variant, right? Because the important thing is the variant is some variants had the capacity to escape the immunity for the vaccines. And this is very important to consider. And also the duration for the immunity and the boost vaccination, we really explained before to some countries, they consider the 3 dose, for example, on the immunosenses is not the same to young people to, for example, for older people they don't have the same immunity. And also the universal vaccination. So when this, I want to say to all the people, they need to receive the vaccine when they are candidate for, and when we had the results with a publication, for example, young and old people in the world. So basically here I show to the vaccination is we need to have all the world vaccinated. For this reason, the COVAS, this is the COVID-19 vaccine global access. They working to have the equitable distribution for the vaccines in the world. For example, here, this is for today. For example, I can show what is the countries to receive the vaccine in percentage in the world. You see here, the more countries to receive more dose on the percentage is people to have high rich, the high economy, or the people to have or to make the vaccine, for example, China, US, European Union. But you see, for example, some countries, they receive less to 10%, for example, continent, African continent. And I want to say, for example, when this phrase to when the pandemic go very fast moving, and everyone is not safe. I mean, no one is safe unless everyone is safe. For this reason, we need to looking for vaccination, but in all the world, and not concentrate only in rich people, in rich countries, because right now people concentrate in receive 3 doses, 4 doses. Well, not is I exactly, it's not really 4 doses, but maybe in the future they consider more booster versus other people today don't have any vaccine. I think I explain. And here is I think it's not clear in this slide, because right now is 47.9% for the world population to receive at least 1 dose in the world. They received 6.7 billion doses have been administered globally. However, only 2.8% of people in low-income countries have received at least 1 dose. So this is something to consider, because you see how it's concentrated. For example, in the blue is the high income, after is upper income. But for example, in many countries today don't have the recourse. They receive 3% for the dose. You see here, for example, in this example, United Arab Emirates is almost all population receive the dose is 96%. But you see, for example, Spain is very good job right now, because it's 81%. This is the case for Mexico. This is 53% to receive at least 1 dose, but only 39% when escape when a fully vaccination. So you see, we need to increase or accelerate the vaccination, especially in young people. Here is only to show, for example, Israel. This is very example when the beginning when they start to vaccinate very, very faster. And several studies, for example, this one published in New England Journal of Medicine, they show this is very important because the efficacy is very high. For example, to avoid the symptomatic case was 95%. and for pass away, for avoid for pass away is 97% versus vaccination versus no unvaccination is, and the study was performed in more to 596 100 people. Other thing to consider is, I explain very quickly. Some studies really show to what is important to vaccinate the majority of the people. For example, I can say very easy example. For example, in one house or in one home to live, for example, 20, maybe it's maybe 10 person in one house. Maybe one person is not possible to vaccinate because for maybe they have problem in the hair, or maybe they had cancer, or maybe had some problems to, it's maybe better to not vaccinate. Another person to not receive the vaccine because maybe he's had 4 years old. Okay, we had 2 people to don't receive the vaccine, but for example, the rest to the people, 8 people receive the vaccine. It's more difficult to these people to they don't receive the vaccine, have the virus, or they receive the virus or transmit the virus, versus the people only receive not 8, only 1 or 2 receive the vaccine. So the several studies show to people to have vaccine in the close contact protect indirectly. I don't know if I explained correctly, but this is the reason for say, okay, it's better to have 90% to people vaccinated, 80%, and with this we can lose the possibility to adhere the virus. And I said before in the beginning, the adverse events, this is the more no minors, this is the more very low, very few commons. For example, this is other problems, for example, Guillain-Barré syndromes, or thrombosis, or myocarditis, or anaphylaxis, but it's very few cases. For example, I show only here is 3 cases for every 1 million the doses. So basically is not very common. So it's more common to you have this thrombosis or this myocarditis if you don't receive the vaccine and you are get the natural infection with the virus. Okay, I go. Other things important is the reinfection. We show to less to people, 0.1 people fully vaccination population. Post-vaccine infection are less asymptomatic. People to have the vaccine normally clear the virus more faster compared to the people to not receive the vaccine, and less to 1% to the people to have the vaccine die, but also is because they had other problems, comorbidities problems, for example, cancer, or they had, for example, 8 years, 2 years old. Here very quickly, this is, I can show the structure, for example, for one variant. This is the Delta variants. I show here some mutation to, this is important, for example, for escape for the virus, for the vaccine, or to interact or to bind to win the ACE2 receptor. Here I can show how is the variants divide. For example, in Mexico, the WHO organization, they divide the variants in 3 groups, in variants of concern, variant of interest, and variants under monitoring. You see here, the more relevant is the concerns, so is the Delta variants. Okay, Stephanie, before I forget.
I was going to tell you that Stephanie has a question. I don't know if you want to do it, Stephanie.
Lo siento, fue sin querer.
Oh, okay, sorry. Okay, we can continue. Okay, the more important is the virus on concern. For example, here is the Delta variants. I can explain everything for the timing, but you see here, I can show how many countries detect the variants, when was identified first, how many times was identified in Mexico, and for example, in how many states also they identify these variants. Here is the graphs. How is the distribution for these variants recently? So you see, for example, the Delta variant is divided for several lineages. And here, the last identification you see is more to 95%. The cases was responsible for the Delta variants. Here is all the Delta variants for the last lot identified. And this is for the Mexican Republic, but also for the state is these graphs, and it's very similar, for example, for Jalisco, right? Another thing to consider is to, I say before, not only the S protein is responsible for the immunity. Several studies in the world show to, for example, the envelope protein, membrane protein, nucleocapsid protein, or other proteins. They have the role to produce neutralizing antibodies or cellular response. With that, we want to say maybe in the future we generate new vaccine to other targets. Here, I want to say, because sometimes you say, okay, this is new variants, for example, Delta variants, new variants, and other variants. Maybe the vaccine is not working anymore, but it's not really true. We really test for several groups and publish to the variants. All the vaccine is worse when all the variants. Only low the efficacy. For example, you see the neutralization antibodies is low compared to the, for example, for the alpha variants, so is coming in the beginning for the Wuhan virus. In all the case, this is for convalescent people, and this is for the people to receive the Pfizer, or this is the people to receive the Moderna vaccine. And also here I show what is important, the age, because as I say before, it's not the same immunity. Sometimes young people have better immunity compared to the old people. I show here, for example, for the variants, you see, for example, is reduced their neutralizing antibodies when the people had 40 years old or versus 60 years old. But also it's important the timing. It's not the same, the virus neutralization 25 days after the administration the vaccine or more to 100 days. This is the reason for many vaccines reduce the neutralization antibody after 6 months or after 8 months. And this is the reason for several companies, or for example, the FDA or the EMA, not Covide right now, but they consider the extra boosts, so the 3 doses. This is a summary here. Reduction of neutralizing antibody will increase the age, neutralizing antibody decrease with the time, and could be unnecessary a third dose or vaccination in older people. Here we need to discuss about for the 3 doses, but here is more, I consider, another college consider to really is more important right now to people today don't receive any vaccine, so any first dose, receive the first dose versus the 3 doses. But it is a ethics discussion. And also I show here one a study about for the CanSino vaccine. They show, for example, here a comparison with Pfizer versus CanSino. And we saw here the first dose with Pfizer, they had 80.3%. With the neutralizing, and the second dose is also approximately 100%, but when the first dose, the CanSino, is 88.89%. I want to say here, I think maybe many people know, to CanSino, they don't really publish so far. The 3 phase for the publication for the 3 phase, but also the company recently said to after 6 months, they start to decrease the neutralization antibodies, and maybe they need to consider it a booster after maybe 6 months. But for the moment, they don't really say more data. And also more faster here is only to show to not only is important the antibodies, also is really important the T cells. And also the T cell is responsible for the efficacy for several variants. You see here, for example, the Brazilian variant or South African variants. And here there's something I want to show in this slide is because it's very important to receive the first dose and the second dose, because, for example, you receive only the first dose, you had low efficacy. If you receive this complete dose, you had the more higher. And also is something to I say here is to the Pfizer, Moderna, AstraZeneca, Janssen, Novas, and other vaccine, they had efficacy with alpha, beta, gamma, and also delta. So it's not really any variants is not working with the vaccine. All the vaccine work with all the variants so far. And the more I think this is my last slide. Something to the company's work is to generate new generation vaccine. For example, this is a sample to how Moderna company work to make new generation for the vaccine. For example, they use 6 mRNAs against 3 different respiratory vaccines in one vaccine to protect influenza virus, respiratory syncytial virus, and also SARS-CoV-2 virus responsible for COVID-19. We cannot explain everything, but this is all the combination. And finally, so far, they had these results in mice, and all the combo vaccines induce robust antibody response to all components in the mice. I say maybe 1 or 2 years after, we produce the vaccine more specific. I can explain more. For example, this is the S protein sequence. All the vaccines they make with the wild type sequence for the S protein, but maybe in 1 year, we check what is the variants, what is the sequence to circulate in the countries, and they made this specific, they made this protein when include the several mutations to circulate right now. What is the future for the pandemic? This is endemic. Also, this depends for different points. The first is vaccine efficacy, effectivities, the evolution of immunity. I mean, how is the new variants coming, and also the animal reservoirs. You know, so I say this is coming for the zoonotic disease, and you see, for example, every time in the news you see is coming new no pandemic, but new epidemics or new virus is not coming before, or this is the reason for saying is emerging virus. For example, Zika, dengue, or another virus is coming every 2 or 5 years. This is, I mean, the conclusion is we need to stay prepared to the next pandemic. And I want to close with this slide. Normally I like to share, because this is one sentence to write the editor-in-chief in the Science magazine, to show to the protecting the world for the severe acute respiratory syndrome and coronavirus 2 cannot happen with an international scientific collaboration. Because it's true, you see in this pandemic, all the people work, especially medical scientists, or nurses, or people to clean their hospital. Many people also university, you know, for example, in Quito, Nala, they start to work with identification the virus, not identification the cases, with how is the program? I don't remember, but you know, when and also they have with the vaccination, you know, the many centers in the U.S. Universidad of Guadalajara, they had the several centers to vaccinate, but this is only the one I want to share for today. Thank you very much for your attention, and thank you again for the invitation. And I am very happy to apply any question to you have. And also thank you to Carla Nuno for invitation.
Thank you so much, Doctor. I think it was a really interesting explanation, everything about the vaccines. Guys, I don't know if any one of you has a question. You can raise your hand, or you can open your microphone, or you can write a question down on the chat and I can read it to the doctor. Remember, these webinars are for practicing our English. You can open your mic. Don't be shy. I mean, it's difficult to speak English, not only for you, it's for us too. So that's why these webinars are for practicing. You can practice also you writing on the chat, or even right now you're listening, no? Well, while you think you have one question, Doctor, I would like to ask you 2 questions that I have. Well, the first one is not a question. The The publication that you had, you can share it. The one that you own. I don't know if you can share it after.
Yeah, which one?
One of the first slides is when it's the description about all the vaccines and even the symptoms and all the stuff. I think it's really interesting for us. So if you want to share with us, it would be great. The other question that I have is, I don't know if you can explain us how, I don't know, it's the nature, it's not your name, the one I'm watching. That one, yeah.
Oh, okay. Sorry, continue.
Yeah, that one.
But what is the question?
The question is, I don't know if I can explain it, but how do you choose the mechanism of the vaccine? I mean, how do you choose if one vaccine is gonna be with a protein or another vaccine is gonna be with a vector or with mRNA? How do you choose that? Or how do you make the selection to have a good vaccine?
Yeah, this is very dependent for the company, basically, because, for example, and dependent for the lab, of course, because in the previous lab, we made light attenuated vaccines, for example, in Spain. But after, for COVID, we are working in US with DNA vaccines. So, but what was the reason first? Because in the lab is sparse in light attenuated vaccine, and we decide to work in light attenuated vaccine. Right now in DNA, because for DNA vaccine is more easy for say something compared, for example, for light attenuated or for inactivated or for other. This is the reason for why say DNA. For example, for mRNA, why? Because RNA, you know, right now is very new technology. And this is it's considered very easy to make the RNA vaccine, for example, the platforms. And this is the reason for, say, for mRNA. Another reason is for, how you say, for the immunity, because they had more immunity compared to other one, but other is the patents. Because, for example, the mRNA vaccine, not all the people can work because, you know, for the patent is the same to the drugs. But this is the special reason. I think the more important is the company to decide how according to what they have experience. Because, for example, other platforms to make more easy vaccine is the protein vaccine, because they only produce in a scale with a company. They produce a lot of protein and they use house a vaccine, but this is not very too complicated to make it. So this is, I think this is the special reason for that.
Okay, thank you so much. In another slide, you said about the stability of the vaccines. And one of the things that you mentioned, it was for the stability, you could reduce the GC nucleotides, right?
Yeah.
Why is the reason? I mean, it's kind of that, I don't know why do you do that for better stability?
In here, right?
I think it's gonna be in the next. Yeah, that one. Reduce amount of GC.
Yeah, it's because they compare in the in vitro how is more stability when TA or when GC, and they saw in vitro to when they had more GC, they had more less stability, and this is the reason for reduce the GC. Okay, I think I don't reply correctly because you asked me the mechanism. I don't really, I cannot help you in this question because I don't really know what is the mechanism for what is the reason for when they had more GC, they had less stability.
Okay, no worries.
I don't really know what is the mechanism.
We have one question on the chat. I don't know if you want to read it and answer it, please.
Yes. I want to know if you can explain more about the effectivity of the Sinovac vaccine, please, and thanks. But more about the effectivity. I mean, every vaccine had a different effectivity. I mean, the Sinovac, Sputnik, Pfizer, something. This is vaccine to is make it for inactivate virus. And they made the name is CoronaVac. But I don't know why you want to explain more about the effectivity. I mean, the effectivity is for avoid the hospitalization and passaways is very high. They had less effectivity, for example, for asymptomatic compared with Moderna or with Pfizer. But always when I explain, for example, the difference versus vaccine, here something too important to say is to all the vaccines, they are good to protect to the people don't die. This is the more important things. That if you want to say this Sinovac had something, this CanSino had less, this Pfizer had more, if AstraZeneca, this is not really too important because, we need to remember to this is a pandemic and the pandemic, the more important thing is to everyone had any vaccine right now. Maybe in the future, maybe in 2 years, in 3 years, okay, we are select what is the best vaccine in the world, and we have produced only this better vaccine. But right now, this is not really too important to say the more difference to the vaccine. I know this is, if you want to know more specific every vaccine, I can share the publication for any vaccine to you want it. I can write my email is you want more specific data. But for the moment, I can say, yeah, they had a little bit less efficacy, for example, with mRNA vaccine, but the more important thing is all the vaccine works with the people don't die, basically.
Thank you so much, Doctor. Thank you, Brenda, for asking. I have one more question. I don't know if anyone has one. If no, I'm going to ask. What did you think about mixing the type of vaccine? I mean, is there any study of the results or what?
Yeah, personally, actually today I had one interview about this one, and also it's very frequent question. I can show also the note to what's published today about for the combination. But in summary, for the moment, it's very few studies to show combination for the vaccine. The more advanced studies is in Spain, also in England, and also in US. The more vaccine to study for combination is Pfizer and AstraZeneca, or vice versa. And also recently in US, they studied a vaccine today make or they administrate the Pfizer, Moderna, and Johnson & Johnson. In general, for the moment, the results show okay, I can explain two terms or two words. Heterologous is when the people receive different vaccine, and homologous is when the people receive the same vaccine. For example, the first dose for Pfizer and the second dose for Pfizer. In general, heterologous vaccination is a little bit better in neutralization percentage, and they don't show significant difference for effect adverse. But something important right now to any countries for the moment recommend combination vaccine in public population. All these studies is for publication or for research to know how it works. in the future, for sure, especially in Euro Union or in US, I don't have, sorry, I don't know how it's say in English. I don't know right now, but no tengo duda. To in the future is possible, and this is recommend to combine vaccine, but not for right now. So we really understand the question, because, you know, especially many people to receive some vaccines today start to see in the news to maybe lost the efficacy after some months, and you know, or some vaccines to the beginning was only considered to the one dose, but after they say maybe we need a boost, for example, you know, Johnson or Johnson. But in general, these things, and also for the coffee price, is not approved anything about that for the moment. And also the FDA, also the EMA. But in summary, yes, it's coming new results about that. And I think the next year is maybe it's possible. And something I want to say right now is, for example, the reason for start to have the combination studies is because the distribution, because, you know, is sometimes it's difficult to have, you know, right now in Mexico, what is the situation? Is no a lot of vaccine. And maybe imagine to Pfizer, they don't produce for any reason million the dose right now. Okay, we can say, okay, right now I had 1 million the people to receive one dose for the Pfizer, but the company, they don't produce right now. Okay, AstraZeneca is fine. We can have the second dose for AstraZeneca. This is a special reason when it start to have these studies. And also, it's not really new to have the heterologous vaccination. For other diseases, it's very common to have heterologous vaccination. But this is very good question. And I want to say to any question to you really had about for the COVID-19 or vaccine, I am very happy to reply. Right now, if you want to write in the chat, it's fine. Okay, I think we had one question. I have one question for Minerva. What do you think about the 3 Pfizer dose? Do you think it's very necessary? Thanks for your response. Yeah, this is the thing too I want to say. Also, this is frequently this question, because in general, okay, I want to say something very important. The 3 dose is not really recommended to general population right now. It's only recommended for some groups for the people, okay? Because sometimes the news, they don't share the title very correctly. But right now, it's only for people to have more to 60 years old, or people to have problems with the immunity, for example, immunocompromised, for example, when have HSV or lupus, or problems with cancer or organs transplant. I don't know if we said correctly, and some these groups, but more for general population. This is about for the 3 dose. So it's different, for example, for a boost when Johnson & Johnson was only 1 dose in the beginning, but we really know now to after 6 months start to reduce. So right now for US, they say to people to receive Johnson & Johnson after the 2nd month, it's okay to have the booster for this vaccine. By my opinion, my personal opinion about for the 3 dose, another opinion to share is similar to other college. I think for me, it's really more important to share these 3 doses when people today don't have any dose, especially in poor countries, because I cannot explain in a slide because for the timing, but really the variant, why is coming the variants? Is coming when the countries, for example, to have very few but percentages for the vaccination. Because how is the variant is coming? Because it's coming when the virus replicate, and the virus replicate in the people normally when they don't have the vaccine, they replicate more faster and faster. And this is the reason for have more variants. And if you had more variants right now, for the moment, thank you, but it's not really lost the all efficacy for any vaccine. only reduce, but maybe in the future, if the people don't share the vaccine, for example, the rich people, maybe they had one variant to escape completely to the vaccine. So for this reason, we say, this is also the FDA. This is the reason for don't really approve the 3 doses in the complete population, because this really is more important to vaccinate more people versus 3 doses. Because this is right now, the majority of the people to go to the hospital is people who don't receive any vaccine. Is right now the pandemic is for the people they don't have the vaccine. Is unvaccinated people. Is around 95% to the people to come into the hospital is people who don't receive any vaccine. This is really my opinion. For the moment, it's not really important because they checking several people to work in immunology. They checking what is the timing to have the people protection. And also for now, 8 months is normally the people to have received the first vaccine in the world, 8 months to 1 year. They also protection antibodies, T cells, and long memories. So when this reason is not really too necessary to have 3 doses in general for the moment, especially if in young people.
Thank you so much, Doctor. I think it was really clear. Thank you, Minerva, for making the question. I don't know if anyone has more questions for the doctor. I think it was really, really interesting, and we could understand a lot of things that maybe we don't catch it when we read it. And was a great explanation for everything, immunology, and even the political situation now about the vaccine. It's a really frame that we don't have that perspective, but we must too. So guys, if you don't have a question, I would like to thank Dr. Angel because it was really, as I told you, really interesting. I know you're really busy. You had a class before this, and I know you were really rushed. But really, thank you. I want to really thank you because it was really, really interesting. Thank you so much, Doctor, for being here tonight. And thank you so much, everybody, for coming to this webinar, our last webinar. Thank you so much, Doctor.
Thank you. Thank you.
So I hope everybody has a great night, a great dinner, and thank you so much. I hope I can see you next semester with another webinars in English. Thank you so much, everybody. Bye.
Bye-bye.
Bye.